Ladies and gentlemen, thank you for standing by, and welcome to the Reata management call to discuss positive top-line pivotal MOXIe data. At this time, all participants' lines are in a listen-only mode. Please be advised that an audio recording of today's webcast will be available shortly after the call on Reata's website at reatapharma.com in the Investors section. If you require any further assistance, please press star then 0. Before the company proceeds with its remarks, please note the forward-looking statements on today's call. There are many factors that could cause results to differ from expectations, including those noted in the company's SEC filings. Today's statements are not guarantees of future outcomes. Please also note that any comments made on today's call speak only as of today, October 15th, 2019, and may no longer be accurate at the time of any webcast replay or transcript rereading. Following the prepared remarks, we will open the call up for questions. We do ask that you please limit yourself to one question and one follow-up so that we can accommodate as many questions as possible. I would now like to hand the conference over to your speaker today, Vinny Jindal, Vice President of Strategy. You may begin.
Thank you. Good morning and welcome to Reata Management's call to discuss the results from the pivotal MOXIe trial of Omaveloxone and Friedreich's ataxia. Yesterday afternoon, we issued a press release with a summary of these results, and the press release, as well as an 8-K filing with the slide deck being presented this morning, can be found on the Investors section of our website at reatapharma.com. I'm joined today by our CEO, Warren Huff, Chief Medical Officer, Colin Meyer, and CFO, Manmeet Soni. I'll now turn the call over to Warren.
Thanks, Vinny. Good morning, everyone, and thank you for joining us. Yesterday afternoon, we announced positive top-line results from the pivotal MOXIe study of Omaveloxone, or Omav, in patients with Friedreich's ataxia. MOXIe was the largest global interventional study ever conducted in FA and was the first to demonstrate a statistically significant improvement in neurologic function in patients with FA. This would not have been possible without the commitment made to this program by the patients, families, and caregivers who live with this disease, and in particular the efforts of the Friedreich's Ataxia Research Alliance, FARA. Today is a truly historic day for the FA community and for our team here at Reata. I'd like to begin today's call by providing some background on Friedreich's ataxia, which is a debilitating, degenerative neuromuscular disorder that profoundly affects patients' motor function and in almost all cases shortens their lives. FA is an ultra-rare orphan disease that affects approximately 22,000 patients worldwide and approximately 5,000 patients in the United States. Patients are normally diagnosed during childhood when they begin to stumble or display other overt signs of impaired coordination. Over time, they become dependent on walkers and then wheelchairs, and they ultimately lose their independence altogether. Unfortunately, the median survival for this disease is in the mid-30s. Until now, Friedreich's ataxia has proven intractable to treatment. A number of interventional studies have been conducted in FA, and none have demonstrated a significant therapeutic effect on the progression of this disease. As a consequence, there are no FDA-approved therapies for patients with FA. Friedreich's ataxia is a disease of mitochondrial dysfunction. The underlying cause of FA is a mutation in the gene for frataxin, a mitochondrial protein that helps assemble the iron-sulfur clusters that are necessary for the production of cellular energy in the form of ATP. In FA, mitochondrial respiration is suppressed and energy production is dramatically reduced. This affects the function and viability of nerve cells. As depicted in the graph here, these impairments manifest clinically as a worsening in neurologic function that correlates with reduced whole-body respiration. Friedreich's ataxia researchers have established that NRF2, the target of Omav, is suppressed and NRF2 signaling is impaired in FA patients. This contributes to mitochondrial dysfunction and reduced cellular energy production. Based on this, we tested whether Omav could restore mitochondrial function in FA patient samples ex vivo. As you can see on the left side of this slide, Omav was able to restore mitochondrial function in cells harvested from FA patients. Since mitochondrial function correlates with neurologic function, we decided to proceed with our clinical program in FA. We initiated the dose-ranging Part 1 of MOXIe to determine if the effect we observed in patient samples could translate clinically to patients, as well as to assess Omav's safety profile in FA patients. Patients treated with the optimal dose of Omav experienced a substantial improvement in neurologic function as assessed by the Modified Friedreich's Ataxia Rating Scale, or mFARS, after just 12 weeks of treatment. The mFARS is a neurologic exam used to track the status of FA patients. Importantly, we also gained insights into the disease and how its progression is measured, which helped us design the pivotal Part 2 portion of MOXIe. We made a few important adjustments to the study that included increasing the sample size substantially, extending the duration of treatment to 48 weeks, and narrowing the primary analysis to patients without pes cavus. Pes cavus is a foot deformity that may interfere with the patient's ability to complete the mFARS exam. With that background, I'll turn the call over to Colin to describe the design of the Part 2 of MOXIe in greater detail and to review the results of the study.
Thanks, Warren. The pivotal portion of MOXIe was a double-blind, placebo-controlled, randomized international study that, as Warren mentioned earlier, was the largest global interventional study ever conducted in FA. We enrolled 103 patients across a wide and representative range of age and disease severity. Patients were randomized 1-to-1 to receive 150 milligrams of Omav or placebo for 48 weeks. The primary analysis population included the 82 patients who did not have pes cavus, and the primary endpoint was the change from baseline in mFARS scores at week 48. Safety analyses were evaluated in the all-randomized population of 103 patients. As Warren noted, the progression of FA is measured in clinical studies by the Modified FARS or mFARS scale, a physician-assessed neurological exam. It is comprised of 4 sections that reflect the patient's ability to perform everyday tasks. The bulbar section primarily measures speech and swallowing. The upper limb section measures the ability of patients to use their arms and hands. The lower limb coordination section measures the ability of patients to use their legs. And the upright stability section measures the patient's ability to stand and walk. The mFARS accurately tracks disease progression in FA patients, and as patients lose function, their mFARS score climbs on average 1 to 2 points annually towards an upper limit of 99 points. Most patients are diagnosed when they have a score between 25 to 35. Once their scores reach approximately 55 to 60, they become wheelchair bound and can no longer walk. And patients unfortunately die once their scores approach the upper end of the range. The FDA provided us with guidance that mFARS is an approvable endpoint in FA, and we defined mFARS as the primary endpoint of the trial for MOXIe. MOXIe successfully met its primary endpoint of change in the mFARS relative to placebo after 48 weeks of treatment. Patients treated with 150 milligrams of Omav demonstrated a statistically significant, placebo-corrected 2.4-point improvement in the mFARS compared to placebo after 48 weeks of treatment, with a p-value of 0.014. Patients treated with Omav experienced a mean improvement in mFARS of minus 1.55 points from baseline, while patients treated with placebo experienced a mean worsening in mFARS of positive 0.85 points from baseline. Patients treated with Omav for 48 weeks regained an amount of function that is typically lost in the course of 1 year. Moreover, in the all-randomized population that includes patients with pes cavus, Omav treatment improved mFARS by 1.93 points relative to placebo, which was statistically significant with a p-value of 0.034. The change in mFARS over time in both treatment groups is shown on slide 9. As seen in virtually all prior FA trials, the placebo-treated patients had an initial improvement. This effect was lost over the following 36 weeks, and by week 48, these patients had worsened by 0.85 points. These data are consistent with available natural history data for FA patients that was published in 2016 and demonstrated an annual worsening of approximately 1 to 2 points.
In contrast, patients receiving Omav improved from baseline and maintained their improvement through the 48-week treatment period. Importantly, the largest placebo-corrected improvement was observed at week 48 at the end of the study. A paper was published in 2016 by Patel and several FA KOLs, including 7 of the 11 investigators in MOXIe, which quantified the rate of progression in FA over 5 years. They performed power calculations and estimated that 545 FA patients would be needed to adequately power a 1-year trial to demonstrate a difference in mFARS. Our trial was successful because the treatment effect was greater and demonstrated not just slowing of progression, but improvement in the Omav-treated patients from baseline. In addition to the change in mFARS, several secondary endpoints were included in MOXIe and analyzed hierarchically. They were included to descriptively characterize specific functional characteristics related to the disease. Several of the secondary assessments, including the Activities of Daily Living Scale, or ADL, were patient-reported outcomes. These were included to determine if changes in neurological function, as determined by the mFARS and assessed by the investigator, were associated with changes in the way that patients reported how they felt or functioned. It was not feasible to power the trial for these endpoints. For example, Patel and colleagues estimated that over 2,000 patients would need to be studied for approximately 2 years to show a difference in the ADL scale. As we expected, a numerical improvement was observed in the key secondary endpoint, the Patient Global Impression of Change, with a p-value of 0.12. In the all-randomized population, this assessment demonstrated nominal statistical significance with a p-value of 0.028. In both analysis populations, a higher proportion of Omav-treated patients reported improvements, and a lower proportion reported worsening than the placebo-treated patients. Notably, the scores in this patient-reported outcome correlated significantly with the change in physician-assessed neurological function. The ADL is a patient-reported outcome that measures 9 everyday aspects of life with FA, including speech, swallowing, use of hands to cut food, dressing and performing personal hygiene, falling, walking, sitting, and bladder function. The ADL demonstrated nominal statistical significance with a p-value of 0.042. In all 9 questions, the ADL demonstrated numerical improvements relative to placebo. Because patients with FA progressively lose the ability to coordinate their legs and walk, they are prone to falling, which is obviously dangerous and limits their independence. As further evidence of improved coordination, patients on Omav fell 65% less than placebo patients over the 48 weeks of the study. The improvements in the secondary endpoints, including the Patient Global Impression of Change, ADL, and frequency of falls, suggest that Omav may be having an impact on these patients' daily lives. These patient-reported outcomes support the significant improvements in neurological function as determined by mFARS and assessed by the investigators. Regarding safety, Omav was generally reported to be well tolerated, and reported adverse events were generally mild to moderate in intensity. Only a small number of patients discontinued due to adverse events in either arm. Notably, 98% of eligible patients elected to enroll in the extension phase of the trial. The most common adverse events that were reported in greater than 20% of patients are shown in the table. Those that were observed more frequently compared to placebo were headache, nausea, increased aminotransferases, fatigue, and abdominal pain. These adverse events generally occurred shortly after initiation of Omav treatment and were reduced in frequency after the first 12 weeks. Increases in aminotransferases are a pharmacological effect of Omav, which increases production of aminotransferases in vitro, and which we believe are related to the restoration of mitochondrial function, because these enzymes play an important role in cellular energy production. In support of this, changes in ALT at week 4 in MOXIe significantly correlated with improvements in mFARS at week 48. The aminotransferase increases were associated with statistically significant improvements or reductions in total bilirubin and were not associated with liver injury. The overall rate of cardiac and vascular adverse events was low, and fewer Omav-treated patients reported these AEs. The overall rate of serious adverse events, or SAEs, was low, with 3 patients in each group reporting SAEs while receiving study drug. 2 additional Omav-treated patients reported SAEs approximately 2 weeks after receiving their final dose. No new safety signals were identified, and the reported SAEs were sporadic and generally expected in FA patients. In the 3 Omav patients who reported SAEs while receiving Omav, none led to discontinuation. Atrial fibrillation was balanced and reported in 1 Omav and 1 placebo patient. 1 Omav patient reported anemia that was due to a complication of a procedure and was considered unrelated to Omav. 1 Omav patient reported multiple SAEs, including viral upper respiratory tract infection and laryngitis, along with palpitations, non-cardiac chest pain, and sinus tachycardia. While several of these SAEs were considered possibly related to Omav, no imbalances, infection, or arrhythmia adverse events were observed overall in the trial. Notably, over the course of the trial, the Data Safety Monitoring Board met quarterly, reviewing all data on an unblinded basis, and did not note any safety concerns. With that, I will turn the presentation back over to Warren.
Thanks, Colin. In summary, we believe that the results from MOXIe provide significant evidence that Omav produces a clinically meaningful benefit to patients with FA, and we believe that it has the potential to become the first FDA-approved therapy for this disease. We plan to work closely with FARA, who has been an excellent partner, to communicate with the patient community, and we're planning to meet with the FDA and other regulatory agencies soon to discuss the appropriate next regulatory steps. As we look towards a commercial launch of Omav, we are currently assessing the feasibility of launching an early access program in the United States. In parallel, we will continue and accelerate the launch and preparations that began over a year ago, and we now plan to extend these activities to include the territories outside of the United States that we recently reacquired. I'd like to make one final point about the MOXIe results. We believe that the results are also important because they provide proof of concept that improvements in mitochondrial function from Omav treatment may provide a therapeutic benefit in a number of other neurodegenerative diseases. The processes of neuroinflammation and impaired energy production that NRF2 activation can resolve occur in other regions of the brain, causing other rare and debilitating neurodegenerative diseases. We've studied Omav and related molecules and have observed promising preclinical results in models of Huntington's disease, ALS, Parkinson's disease, Alzheimer's disease, and epilepsy. We plan to develop Omav clinically for one or more of these diseases. With that, I'd like to open the call to questions. Operator, please begin the Q&A.
Thank you. And as a reminder, to ask a question, you will need to press star then 1 on your telephone. To withdraw your question, please press the pound key. And as a reminder, we do ask that you please limit yourself to 1 question and 1 follow-up. And our first question comes from Yigal Machamovitz from Citigroup. Your line is open.
Hi, congratulations on this outstanding result. I just had a few questions with respect to the FDA filing for Friedreich's ataxia. Just first of all, a 3-part question. Can you confirm that this study will be sufficient to follow with and that you don't need any other clinical work? And do you believe that you'll be able to get a label encompassing the non-pes cavus as well as the pes cavus patients? And thirdly, is it your expectation that the secondary endpoints related to activities of daily living and the patient global impression of change are aspects that you believe could go in the label? Thank you.
Hi, Yigal. So we'll need to obviously meet with the regulators to determine if this information will be sufficient, within the pivotal portion of MOXIe for approval. FDA obviously commented in writing that MFARS could serve as a basis for approval, either accelerated or full approval, and that determination will be made on the basis of the data, which in our view is quite strong. Based upon us showing activity in patients with pes cavus, we think it's likely that those patients would be on the label. Obviously, the FDA will need to make that determination, but not only did we hit the ITT, the primary analysis for all patients, we did show numerical improvements in patients with pes cavus. And thirdly, as far as the other secondaries, and if they're on the label, that will be up to the FDA to decide. But we think the data are very strong and support that the physician-assessed neurological function is associated, and in some of these cases does correlate with how patients report that they felt and function. And so we think those are critical aspects of the overall data package.
Okay, great. And then just one follow-up. Warren, you mentioned some of the indications you might pursue post Friedreich's ataxia. Of the ones you mentioned, is one of them more connected to NRF2 biology versus the others among Huntington's and ALS and some of the others you referenced? Thanks.
No, not really. They all have as a strong feature significantly impaired mitochondrial function. And I think biologically the setting is really similar to what we see in chronic kidney disease, where various forms of diseases have a different initiating insult, but they end up activating inflammatory pathways that impair mitochondrial function and lead to chronic inflammation. And so today it would be difficult to determine exactly which one would be better than the other, just based on the biology. So we'll be triaging them based on, regulatory endpoints, unmet need, and other factors like that, feasibility of the study.
Great. Thank you very much.
Thank you. Our next question comes from Joseph Schwartz from SVB Leerink. Your line is open.
Hi, good morning, guys. Thanks for taking the question. This is Dagon dialing in for Joe, and let me add my congrats on the outstanding data as well. So maybe one for Colin and one for maybe Manmeet. On the mFARS benefit, I was just wondering if you could provide a little more granularity. So, for example, what was the mean baseline of mFARS of the enrollment? I think Part 1 had a 41-point baseline. So what about Part 2? And with regards to the trajectory, you showed us the curve, but I was wondering if you could provide a little more granularity on what subdomains of mFARS perhaps benefited more, if there was any. And then for Manmeet, on the commercial front, perhaps you could provide a little more color on what operations are underway. I know you mentioned working with FARA to communicate the findings and getting more patients involved, and perhaps on the early access part, but with regards to the reacquired rights to global settings, what work has been done there, and I guess what can we anticipate in terms of OpEx going forward? Thanks.
Sure. So to answer your question about mFARS and so the trajectory and the baseline. So the baseline for the second part, the pivotal portion, was approximately 40 as well. So it was pretty close to the first part of the trial. And just to, I think, to quantify the change, as I mentioned, and so that Patel paper actually that I referenced from 2016 accessed data from a natural history study that's enrolled over 1,000 people. It's extraordinary that FARA and the investigators have been able to enroll over 1,000 patients and follow them for over 5 years. And patients typically progress 1 to 2 points annually. And so we think that 2.4 is quite meaningful. As far as the subdomains, we'll disclose that at a future medical meeting, but obviously within the presentation we showed the components of ADL, and so you can see the 9 different questions, and some of those related to speaking and swallowing, some related to use of hands, and some related to use of legs and standing and walking. And so we think that there is a benefit that was quite broad in these patients.
Yeah, this is Manmeet, and on the commercial readiness, what I can say is, we have been, getting ready with our commercial, infrastructure, and already hired our leadership team. We have our chief commercial officer and heard direct reports on, market access, on marketing side, sales team. All leadership team has been already hired, and now with this data, we will, start planning on the further activities. On your second question on OpEx. I would say we have been extremely efficient until now in managing these 4 pivotal trials over here, and we will continue to be efficient. So you should expect a little bit increase in our OpEx in the near term, but not a significant one until we get approval where we hire our sales force and all of the stuff. But still, since this is an ultra-rare disease and most of the patients, as you know, are covered through 8 centers of excellence, we don't need a huge army of sales reps. So even our sales and G&A expenses should not expect to go much higher.
Great. Congrats again, and thanks very much for taking the question.
Sure. Thank you.
Thank you. Our next question comes from Adam Walsh from Stifel. Your line is open.
Okay, great. Good morning. Let me add my congratulations on great data. I have a few questions. The first one is, it was my understanding that one of the key secondary endpoints was change of peak workload during exercise testing. Was that a secondary endpoint, and do we have the results on that? That's number one. And then number two is, in terms of meeting with the regulatory agencies, do you have any visibility on timing? And after that meeting, how will you announce the outcome of that? And then finally, just if you could comment on the FARA registry, what you know about that in terms of patients identified currently with Friedreich's, both in the U.S. and ex-U.S. Thanks.
Sure. So Adam, peak work was included as a secondary. It was not a key secondary. It was down the list in the hierarchy. Recall that we had included it as one of the key endpoints in the first part of the trial, and we saw no meaningful improvement. We included it in the pivotal portion because we thought it would be helpful to condition the patients. And so in a lot of assessments that are effort-based, you can obviously have variability because of the time of day or the different state of the patient. And so in the pivotal portion, we require patients to do the exercise testing first and then undergo the mFARS testing. And so that was the main reason to include it within the trial. Your second question was? Please repeat.
Oh, yeah, no worries. Just how you'll go about announcing your interaction with the FDA so we can get some visibility on kind of some of the regulatory pathway questions.
Yeah, sure. This is Warren. Yeah, obviously we'll be letting the public know what the FDA response is as we go through our pre-NDA meeting process, because it's obviously very material information.
Okay, great. And then the final one was on the FARA registry. Can you give us an update on what you know about patients identified with Friedreich's in the U.S. and ex-U.S. and your interaction with FARA? Thanks.
Sure. And so I guess first, we've had an excellent relationship with FARA. They were critical to the design of the trial, helping us to work with the investigators and help us to access the patients in the trial. And so even though they're U.S.-based, they work internationally with other ataxia groups and centers of excellence. And so you can actually go to FARA's website, and they have very clear information. And so as of today, there's just under 1,800 patients in the registry. And so those patients are alive. And there's obviously more outside the U.S. And so there's already a reasonable number of patients who are followed out of the prevalence of 5,000 patients that we estimate who are in the U.S.
Great. Thank you.
Thank you. Our next question comes from Maury Raycroft from Jefferies. Your line is open.
Hey, everyone. Good morning, and I'll add my congrats, and thanks for taking my question. So you're seeing clear separation of the curves over time. Just wondering if you can comment on day-to-day variability, and did this decline over time for patients? And will these patients continue to be treated and followed to see if there's additional improvement on secondary endpoints?
Sure, and so the variability was pretty much in line with what we expected. Obviously, the p-value showed a low value of 0.014, and so there's clear separation that takes into account variability. The estimate of the p-value was based upon an MMRM methodology, and so it actually included all available mFARS values, not just the mFARS value at week 48. And so we feel like the mFARS results are robust. And yes, there is an extension phase that we call Part 3, and so patients have rolled over, as we mentioned. 98% of eligible patients in Part 2, the pivotal portion, rolled over into Part 3, the extension phase. We think that's a remarkably high number. and demonstrates the interest from the patients and the tolerability from the drug. And so we will be following those patients, and we will release data at a later point in time.
Got it. And as far as biomarkers go, is there anything including ferritin and GCT worth highlighting? And do you have a specific medical meeting in mind where you plan on presenting the additional detailed data?
Yeah, so I think there are multiple biomarkers that demonstrate activity in these patients. We highlighted some of those in the first part of the trial. We'll disclose them in detail at a future medical meeting and in a future, hopefully high-profile publication. But just initially, NRF2 target genes, ferritin and GGT, clearly went up as expected. Those are transcriptionally regulated by the target of our drug, NRF2. Then downstream markers like ALT and AST, which are fundamental to energy regulation, were increased in a pharmacological pattern that we've seen not only with this drug, but actually with our other drug, bardoxolone, too. Most people are unaware, but ALT and AST, as I mentioned, are fundamental to energy metabolism. They did not evolve to be biomarkers of toxicity. One of the products of the reaction from ALT is pyruvate, which obviously goes directly into the TCA cycle, which is obviously the main component of aerobic respiration that produces reducing equivalents for the mitochondria that are used to produce energy. And so we thought it was actually pretty striking that in a relatively small study, we saw a significant correlation between ALT change early on at week 4 and benefit from mFARS at week 48. So yeah, the biochemical data are consistent with prior studies, and I think paint a pretty clear picture.
Great. Congrats again, and thanks for taking my question.
No problem.
Thank you. Our next question comes from Charles Duncan from Cantor Fitzgerald. Your line is open.
Thank you. Let me add my congratulations, Warren, Colin, Mehmet. Really excellent results. Surprising, nice to see for the patient community. I had a couple of questions. One is, when you consider the sample, do you believe that in terms of mFARS, baseline or even subdomains of mFARS, the sample is representative of the broader patient population in FA?
Yes, we do. And so we actually had very broad eligibility criteria, but we took important measures so that we limited variability so that we could see a signal. And so, for instance, the age range was between 16 to 40, so very large for these patients. And the mFARS range was as low as 20 and as high as 80. And as I mentioned earlier, most patients aren't diagnosed until their scores are 25 to 35, and so it was a very broad range. I think a key aspect of this trial was our insight from the first part in other trials where we really tried to limit variability. And so we took multiple measures, including requiring that patients had practiced mFARS exams before the real tests that were used for eligibility were determined. We took 2 independent tests at baseline. They had to be within 4.5 points. Otherwise, they were not eligible to enroll. And of course, we had multiple mFARS exams during the course of the trial because we expected a placebo effect. We wanted to make sure that the placebo effect wore out and that any training effect was normalized by the time patients entered. And so I think it was broad, but we were able to select for a patient population where we were able to detect a clear signal.
Very nicely designed and conducted trial, Colin. Hats off to you. Regarding data, and I imagine some of this you're going to present later, and so you probably won't answer this question, but I'm wondering if you could provide us any additional information with regard to responder analysis. Were there patients that were kind of outside the norm in terms of their response to Omaveloxone?
Yeah, you're right, Chaz. We're going to defer that answer, and we'll provide additional details at an upcoming medical meeting.
Okay, and then final question is regarding timelines. I know people have asked you a bunch of times, but is this something that you could see an NDA filed within, say, 12 months or something on the order of less than that?
Yeah, of course, I'll qualify this by we need to go interact with the FDA and the international regulatory agencies, and of course then we'll provide guidance once we have decisions from them. But we would expect the NDA filing to be along similar time frames, for other NDAs for rare orphan diseases, like this.
You guys are going to be busy. Congrats on the results. Thanks for taking the questions.
Yeah.
Thank you. Our next question comes from Brian Skorney from Baird. Your line is open.
Hey, good morning, guys, and congrats on the data from me as well. I guess my question is, given the proximity of the repurchase of the bardoxolone and omaveloxolone rights that you announced last week, did you or AbbVie get to see any of this data ahead of completing that deal last week? And can you just kind of review when the data was unblinded to as I suppose neither of you had the data ahead of that deal, how you ensure that there wasn't leaking of the data going into that outcome? Thanks.
Sure. Yeah, it's been interesting to watch the speculation after the announcement, but let me be really clear. Neither AbbVie nor us had access to the results of either MOXIe or Cardinal prior to doing the transaction. We engaged with them a while ago to try to resolve what would happen with the regulatory and commercial rights in their territories, and both AbbVie and Reata were interested in resolving it so that the products could be gotten to the patients as rapidly as possible after data was available. In the case of MOXIe and FA, this is a rare, deadly disease with no approved therapy. We were going to have pivotal data, and it's a similar situation with Cardinal and with Alport syndrome patients. And I think both companies were very sensitive about the need of patients in territories outside of the United States, so we endeavored to enter into a transaction that would be beneficial for both parties. But neither of us had a window into the data. We obviously, on our side, thought that both studies would be successful, and we approached the negotiation that way. And luckily, MOXIe is successful, and hopefully Cardinal will be successful as well. But it very well could have gone the other way.
Thank you. And again, ladies and gentlemen, to ask a question, please press star and then 1 now. And our next question comes from Matt Kaplan from Ladenburg. Your line is open.
Hi, good morning, guys, and congrats on the results. Wanted to just dig in a little bit more into the side effect profile, specifically, I guess, the increases in liver enzymes. In managing those, were there any dose reductions that were needed or incorporated into the protocol to address the increases in liver enzymes?
Sure. So I guess the first comment I would make is that we've shown in numerous different types of cells, so in cell culture, that omaveloxolone as well as bardoxolone increase production of transaminases from multiple tissues. And so while, yes, most people refer to these as liver enzymes, we've shown increases in production in not only the liver, but in the kidney, in muscle, and other tissues. And so it's a transcriptional effect of the drug that across our entire platform has never been associated with liver injury. As I mentioned, in MOXIe, we saw a significant reduction in bilirubin. And obviously, bilirubin is the functional measure of the liver. And so in our view, liver function actually improved. When drugs cause injury to the liver and the liver can't do its job, bilirubin goes up. And so once again, this is clearly a pharmacological effect and not associated with injury. We do have to be cautious, of course, in our clinical trials in case there ever is a potential event of liver injury. And so we do include standard rules that I believe likely all biotech and pharma companies include in their trials. And I believe we had a patient who had to stop drug because they hit a predefined threshold. But that's a patient who likely was having a large pharmacological effect, and there was no evidence of injury in that patient.
Okay, that's helpful. And then second question, in terms of the consistency of improvement, you showed overall improvement for patients, but in terms of individual patients, how consistent was it in terms of observing improvement in the mFARS across individual patients?
So I think we perform those analyses and we'll disclose them at a future time. But yeah, obviously, if it's a robust p-value, if the distribution would not have shifted, we would have been unlikely to see a significant p-value. If it was just a few outliers that drove the change, the p-value would have been unlikely to be significant.
Okay, very helpful. Well, thanks for taking the questions, and congrats again on the results.
Thank you.
Thank you. And I am showing no further questions from our phone lines.
Thank you, operator. That concludes this morning's call. We look forward to updating you again soon.
Ladies and gentlemen, an audio recording of today's webcast will be available shortly on Reata's website, reatapharma.com, in the Investors section. This concludes today's conference. Thank you for participating. You may now disconnect. Everyone, have a wonderful day.